Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article, Identification of a Poor Prognosis BRAF-mutant-like Population of Colon Cancer Patients by Vlad Popovici et al. My name is Jeffrey Meyerhart, and I'm a gastrointestinal oncologist and associate professor of medicine at the Dana-Farber Cancer Institute and Harvard Medical School, both in Boston, Massachusetts. My oncological specialty is gastrointestinal cancers. The title of this podcast is What's Different Between Colorectal Cancer and Another? It's all in the genes. Colorectal cancer is the fourth most common cancer diagnosed in the United States, with approximately 140,000 cases diagnosed annually. It is the second most common cancer globally, with an estimated 1.2 million new cases each year. 50,000 people die of colorectal cancer in the United States, and over 600,000 across the globe each year. As realized for nearly all types of cancer, the molecular changes that occur leading to colorectal cancer are not the same in each patient. While this fact has been realized for multiple decades, more recently it is clear that the changes that lead to cancer developing can impact both prognosis and efficacy of treatments. While stage and simple pathological features of the tumor have driven treatment decisions for patients with colorectal cancer for the past 30 years, we are finally starting to get a deeper molecular understanding of this type of cancer, with the goal of individualizing treatments to a particular patient. However, before this lofty goal can be anywhere near a reality, we need to classify the various permutations of mutations that ultimately lead to the entity known as colorectal cancer. In a landmark science paper in 1990, Eric Fearon and Bert Volkelstein first proposed a model for the transition from normal colonic epithelial to hyperproliferative epithelium to early adenomas to late adenomas to carcinoma and to metastases. Along each of these steps, molecular alterations were proposed. While it was realized that not all colorectal cancers require the same molecular alterations, and since that 1990 paper, many additional chromosomal, molecular, and epigenetic changes have been realized, it is clear that colorectal cancer requires multiple alterations to develop. One early change leading to an early adenoma to a more advanced adenoma is a mutation in KRAS. RAS proteins are small GTP aces that cycle between an active guanosine triphosphate, GTP, bound state to an inactive guanosine diphosphate, GDP, bound state. GTP-bound RAS regulates a variety of cellular processes, including proliferation, survival, and differentiation. RAS activates multiple downstream effector pathways, including the RAF-MEK-ERK pathway and the PI3K-AKT pathway. KRAS mutations are detected in 30 to 40% of colorectal cancers. RAF proteins are serine-threonine protein kinases that are activated by RAS in a GTP-dependent manner. Most RAF functions are mediated by phosphorylation, leading to activation of the mitogen active protein known as MEK. Activated MEK phosphorylates extracellular signal regulator kinases, ERK, which in turn regulates cellular functions, including cell cycle proliferation and cell survival. BRAF is reported to be mutated in between 5 and 15% of colorectal cancers. BRAF and KRAS mutations are essentially mutually exclusive, with rare exceptions reported. Interestingly, BRAF mutations have been associated with MLH1 inactivation that results from promoter methylation. As such, 25 to 60% of sporadic microsatellite unstable high tumors harbor BRAF mutations. Multiple prior studies have demonstrated that activating KRAS mutations are not associated with prognosis in patients with either early stage or metastatic colorectal cancer. In 2006, Levery and colleagues first reported that KRAS mutations are predictive of response to cetuximab therapy in patients with metastatic colorectal cancer. The suggested mechanism of this association is that activation of EGFR leads to phosphorylation of wild-type KRAS, which activates its downstream pathway. Blockage of ligands from binding to EGFR with an EGFR inhibitor to the extracellular domain inhibits this activation of the KRAS pathway. However, tumors that harbor a KRAS mutation will have constitutively active KRAS and the resultant downstream pathways, so blockage of EGFR will not impact on the RAS-RAF pathway. Since that initial publication, many subsequent studies have confirmed the predictive value of KRAS mutations when considering either cetuximab or panitumumab as single agents or in combination with cytotoxic chemotherapies in both first and later-line metastatic chemotherapy regimens. These results have led to the provisional clinical opinion published in the JCO on January 12, 2009, changes in the NCCN guidelines for the usage of cetuximab and panitumumab therapy, and changes in the FDA labels for these drugs. Studies have consistently demonstrated that BRAF mutations are associated with a substantially worse prognosis in colorectal cancer. However, given the association of BRAF mutations with MLH1 inactivation and microsatellite instability, there appears to be two populations of BRAF mutations, those with BRAF mutations and microsatellite stability, and those with BRAF mutations and microsatellite instability. In a recent analysis of the Cancer and Leukemia Group B trial, 89803, an adjuvant chemotherapy trial for stage 3 colon cancer comparing 5-fluorouracil and leucovorin to 5-fluorouracil, leucovorin, and irinotecan in the IFL bolus regimen, Ogino and colleagues reported in Clinical Cancer Research in 2001 that BRAF mutated microsatellite stable tumors have significantly worse overall survival compared to BRAF wild-type microsatellite unstable high tumors, with a 5-year survival of 61% for those with BRAF mutations and microsatellite stability compared to 79% for those with BRAF wild-type and microsatellite unstable high tumors. Tumors that were BRAF mutated with microsatellite unstable high and BRAF wild-type with microsatellite stability had intermediate prognoses. The difference in prognoses were consistent for disease-free and recurrence-free survival as well. Given that BRAF is a downstream effector of KRAS, there were investigations whether BRAF mutations also were predictive of outcomes. These investigations have been less straightforward than in KRAS. Initially reported by D. Nicolantonio and colleagues in the JCO in 2009, presence of a BRAF mutation predicted worse progression-free and overall survival to either cetuximab or panitumumab in the second or later line therapy. However, in contrast, retrospective analyses from 2 randomized trials incorporating cetuximab in the first-line setting, the Crystal trial comparing FOLFIRI to FOLFIRI and cetuximab, and the OPUS trial comparing FOLFOX to FOLFOX and cetuximab, demonstrated that BRAF-mutated colorectal cancers still derived a benefit from cetuximab with statistically significant improvements in response rate and non-significant trends towards improved progression-free and overall survival. Despite the evidence of a predictive value of KRAS and prognostic value of BRAF in colorectal cancer, it is evident that not all KRAS wild-type tumors will benefit from treatment with an EGFR inhibitor, nor do all BRAF mutant tumors behave as aggressively with grim prognoses. This concept returns to the fact that not all colorectal cancers have the same mutational changes in their development, and there are multiple permutations of mutations. As such, we cannot look at one gene at a time to understand the phenotype of an individual patient's colorectal cancer. The paper in this week's JCO by Dr. Popovici and colleagues entitled Identification of a Poor Prognosis BRAF-mutant-like Population of Colon Cancer Patients is an important step forward in this regard. The authors utilize a cohort of 668 stage 2 and 3 colon cancer patients with available formalin-fixed, paraffin-embedded tissue blocks treated on the PETAC3 adjuvant trial comparing infusional 5-FU and leucovorin to infusional 5-FU, leucovorin, and irinotecan in the FOLFIRI regimen. The group constructed a 64-gene-based expression classifier to compare BRAF-mutated tumors to KRAS wild-type BRAF wild-type tumors, referred to as the double wild-type tumors, and to KRAS mutant tumors. The classifier was then validated in 3 independent data sets. The 64-gene classifier had a 96% sensitivity and 86% specificity for detecting true BRAF-mutated tumors in both PETAC3 and the validation data sets. However, the most interesting part of the study was that the classifier could identify tumors that acted like BRAF-mutated tumors in terms of poor prognosis, but didn't actually have true BRAF mutations. These tumors with poor prognosis shared many of the other gene mutation or changes that were seen in true BRAF-mutated tumors. Such tumors were found both in the subset of double wild-type tumors as well as KRAS-mutated tumors. In the paper, the authors present 3 endpoints: overall survival, relapse-free survival, which in PETAC3 is the same as disease-free survival as defined in other adjuvant colon cancer trials, and survival after relapse. Given the nature of BRAF mutations being particularly relevant in terms of prognosis for early-stage patients if they recur, I will focus on survival after relapse. As anticipated, the hazard ratio for survival after relapse for true BRAF-mutated tumors compared to BRAF wild-type tumors was 3.68, with a 95% confidence interval of 2.20 to 6.16. Similarly, the hazard ratio comparing the predicted BRAF-mutated tumors based on the signature compared to the predicted BRAF wild-type tumors was 2.85 with a 95% confidence interval of 2.06 to 3.95. Of interest, within the true BRAF wild-type and KRAS wild-type tumors, those that are true BRAF wild-type but have a BRAF mutated-like signature had significantly worse hazard ratio for survival after relapse of 2.72 with a 95% confidence interval of 1.46 to 5.06 compared to the predicted BRAF wild-type tumors. In Figure 2 of the paper, a host of Kaplan-Meier curves for overall survival and survival after relapse were presented. The curves for the true BRAF wild-type tumors that had BRAF mutated-like signatures were consistently worse compared to both the predicted BRAF wild-type tumors. In fact, the curves from the true BRAF wild-type tumors with BRAF mutated-like signatures were nearly identical to the true BRAF mutated tumors for both endpoints. Where do we go from here? First, the paper presents a strong case for more in-depth molecular profiling of tumors beyond simple single mutations, one at a time. The paper provides insight into why not all BRAF wild-type tumors behave the same. Now the next step is needed. While prognostic, does the BRAF mutated-like signature predict sensitivity or resistance to chemotherapy? Can one use this or other signatures to guide patient care? Guidance can come in many forms. In early-stage disease, can this or other signatures provide which patients should and should not get adjuvant therapy? In advanced disease patients, can the signature direct who should we offer EGFR inhibitors or other molecular targeted therapies, and who should we not offer these agents? Can the signature guide what line of therapy such agents should be included in? 14 years ago, only 5-fluorouracil existed for the treatment of metastatic colorectal cancer. Now there are 7 FDA approved drugs in 5 classes. However, with the exception of KRAS status, we don't know which therapies to use in which patients to optimize efficacy and minimize toxicity or effects on quality of life. Work like that of Dr. Popovici and colleagues will hopefully finally move us in that direction and beyond. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.